Okay, thank you very much. I'm David Simon. As Dr. Shen pointed out, I'm helping with the outreach portion of the Udall Center. I'm also a neurologist and director of our Movement Disorders Center at Beth Israel Deaconess Medical Center. And I'm going to talk to you today about the possibility of a vaccine for Parkinson's disease, which almost sounds like an error in a question, because Parkinson's disease isn't an infectious disease. Vaccines are for infectious diseases, right? So I'm going to talk a little bit about why we're talking about this subject today. I do have some disclosures on the slide. One that's relevant is I'm participating in a steering committee for one of the clinical studies that I'm going to be talking about today, sponsored by Biogen. So what I'm going to talk about today, first I'm going to just describe what is alpha-synuclein. You've heard some of it today, and I'm going to go over a little bit more about what it is and why we think it plays a role in Parkinson's, and why we think too much alpha-synuclein is bad, and therefore are focusing on strategies to get rid of alpha-synuclein. And in particular, although there's a lot of different strategies being used, I'm going to focus on targeting the immune system basically as a tool to help clear alpha-synuclein. There is a problem with it, though. It's inside cells, which can make it a little bit hard for the antibodies, the immune system, to target it, and we'll talk about that. And is there any kind of bad problem? Is it maybe going to create problems by clearing away alpha-synuclein? And there's some interesting lessons that are important to keep in mind from similar strategies that have been used to target a different protein in Alzheimer's disease that I'll briefly talk about. This is, one of my favorite Far Side cartoons, Great Moments in Evolution, and this is my modification of it, where I think we hopefully stand now, and we'll look at it that way in retrospect. There's also this quote from one of the CEOs of one of the companies, Prothena, that's sponsoring a clinical trial for immunotherapy research. In other words, this is a shot at a cure, referring to immunotherapy to target alpha-synuclein. And the question is this just hype? Is this greatly overstating it by somebody who stands to profit from this statement, or is this actually a reasonable statement? And I'll come back to that at the end, and then you can help me decide. I like Dr. Schwarzschild's more entertaining graveyard slide. This is a few trials that have looked very promising early on but failed, and I'm still very hopeful, and then hopefully at the end of this you'll see why. There's a lot of problems with these other agents. For example, with some of them, the precise mechanism by which they work is unclear. The targets may not be known, and where the targets of these strategies is known, the relevance of that mechanism to Parkinson's disease may be somewhat uncertain. And there's different Parkinson's subtypes. We treat Parkinson's disease for most of these trials as one entity, but in fact there may have been only a very small subset of patients in the trials who stood to benefit from that particular drug. And that's why, for instance, in the uric acid trial, Dr. Schwarzschild pointed out we have a way to screen for at least a subset of patients who might be more likely to benefit. But that wasn't true for most of these studies. So in that context, there is one mechanism, alpha-synuclein, that might be relevant not just to a small subset, but potentially to the vast majority of Parkinson's patients. Alpha-synuclein is a core protein component of Lewy bodies. Lewy bodies are these clumps of protein that form inside the cell. That's that pink substance you see right there, right here. Actually, you see these dark pigments here, that's neuromelanin. It's melanin granules just similar to the melanin in skin that makes skin pigmented, and it's within these same neurons that make dopamine. But it was noticed that they have these clumps of abnormal proteins. Early on, we didn't really know what was the main component of those Lewy bodies. And there was a huge family called the contursi kindred that played a major role in helping us understand what that protein was. This is just first an example of what's called a pedigree, a family tree, and where you see the circles represent women, squares men, and solids filled in circle is somebody affected by the illness. And here's a 3-generation pedigree. But here's the pedigree from the contursi kindred. It's just a massive family, and it, as you can imagine, took a lot of work to trace down all the individuals, figure out who was affected and who wasn't. And this was done back in the 1990s, back before we had a lot of the tools we have today. Today we can basically sequence all the genes in every individual in a family, and we can identify a mutation potentially even in a small family. But back then, it required a large family like this to do what's called linkage analysis, where you gradually narrow down the region of the genome that all the affected people share, and then you find out which gene has the mutation, and then do other studies to prove it. And it was a massive effort to do that, and doing that led to the discovery that in that family and in a very small number of other rare families, they harbored a point mutation in the gene encoding alpha-synuclein. So one thing, sometimes we talk about a gene as a disease gene, like alpha-synuclein. Well, we all have the alpha-synuclein gene. We all make alpha-synuclein gene. But for some reason, when there's a point mutation in this gene, almost everybody who gets that point mutation develops Parkinson's disease. We don't really know the function, but it does play a role in synaptic vesicle movement, meaning communication between cells. And a very important discovery was that this alpha-synuclein is a major component of those Lewy bodies. And that's true in the vast majority of patients, even patients who don't have mutations. So alpha-synuclein accumulates in everybody. Now, if we just discovered that first without the genetic family, we might not know if that really plays any role in the disease. But the fact that when a person can inherit a mutation and that causes the alpha-synuclein to accumulate, and then that causes Parkinson's disease, tells us that this really may play a role. And then came another family, or set of families, that was very instructive. And this was here's one example, not as large as the Contursi kindred, but a reasonably large family with multiple generations of affected individuals. And they did that kind of linkage analysis I said, where they traced it down to see which part of the genome was affected. And it came down to looking like the alpha-synuclein gene was the one that was affected. So they sequenced the alpha-synuclein gene, no mutations. And that's puzzling, because looking at it one way told them this was due to a mutation in the alpha-synuclein gene, but looking at it another way said there are no mutations. And additional studies showed that it actually there wasn't a mutation in the gene. The sequence was normal, but they had extra copies of the alpha-synuclein gene. In fact, one of the two copies had a triplication. So in fact, they had 4 total copies of the gene rather than 2 like the rest of us. So they made about twice as much alpha-synuclein protein, and that caused autosomal dominant Parkinson's disease. So that tells us, first of all, it's dose dependent. Some people only have a duplication, not a triplication. They also get Parkinson's disease, but on average a little bit later compared to those with the gene triplication. So what that tells us is too much alpha-synuclein is bad. Even if it's normal alpha-synuclein without a mutation, simply having too much is bad, which is of relevance to all the other patients who don't have alpha-synuclein mutations, but we see alpha-synuclein accumulating. So again, all of this is encouraging to think that maybe clearing away alpha-synuclein, or at least reducing the levels of it in the brain, could be helpful. Could there be problems from reducing alpha-synuclein levels? Well, you can make mice that completely lack the alpha-synuclein gene, so they don't make any alpha-synuclein, and they do have subtle deficits. Here's an example measuring levels of dopamine in the striatum, and in that part of the brain, the mice that don't have alpha-synuclein actually have lower levels of alpha-synuclein, and you can measure some behavioral deficits, subtle ones. So, for instance, giving amphetamines will make the mice move around a lot, but the mice who lack alpha-synuclein don't move around quite so much in response to amphetamines. So these are examples of a couple of subtle deficits. So one way to potentially get around that, and one way that the companies that are focusing on immunotherapy are hoping will avoid these subtle problems that might occur if you completely eliminate alpha-synuclein, is to target not all alpha-synuclein, but only the toxic forms. So alpha-synuclein, when it's initially produced, are just single molecules like you see here on the left, but these molecules tend to join together, and eventually they form what are called protofibrils and then fibrils, and then those can clump together to form insoluble fibrils, and those kind of clumps of insoluble protein are what forms the Lewy bodies. And the thought is that these aggregated forms are mediating the toxicity, not these, what we call the monomers, the single molecule forms. So they created, antibodies are remarkably specific. Your immune system evolved to be very specific and target not yourself, but to target specific invaders, but here it's used to target a very specific form of the protein, only the aggregated forms. And then you'd leave the single molecules that can still serve their normal function and only get rid of the pathological form, in theory. But another problem I mentioned, okay, so the aggregated forms may be the problem, but those aggregates form inside the cells. It's a little different than, say, in Alzheimer's disease, where amyloid, a different protein that accumulates in the extracellular region. Here you have this Lewy body inside the cells. The antibodies, how are they going to get access to that? Do they have to get inside the cells to clear it away? And not clear if they would do that. But then it becomes important, what might initially seem like a different line of research, and that is the fact that there's evidence that alpha-synuclein actually spreads from cell to cell. So cells, it's secreted, it goes out of the cells, and then it's picked up by other cells nearby, and that may be part of the reason why the pathology in alpha-synuclein spreads. And here's an example of some people who got fetal tissue transplants long ago. These were fetal cells that produced dopamine transplanted into the brain of Parkinson's patients. And those were interesting trials. There was, some evidence possibly of benefit, but also significant problems, like some patients developed dyskinesias, even off medications. But the reason I bring it up here is to point out that a couple of the patients passed away many, many years after getting these transplants. And if you looked under the microscope at their brains, some of the dopamine-producing neurons in their brain, some of the transplanted neurons had alpha-synuclein aggregations. They had developed pathology. So it looks like, now these were from fetal cells, so almost certainly they didn't have alpha-synuclein pathology when they were transplanted. It looks like the synuclein pathology within the environment, the brain environment where these cells were transplanted, spread into the transplanted cells. So that's a potential problem when you think about the use of maybe stem cells or fetal transplants. But it's also a potential opportunity when maybe the antibodies can access the alpha-synuclein during this extracellular part of its pathway and prevent the spread. Even if it doesn't clear it away from existing cells, if it stops the spread of the disease, that could clearly be beneficial. So this is a good example of how, at a research end, we go between humans and animal models and back to humans again. So with alpha-synuclein, the gene was first identified in humans. And the genetics in the human families told us that maybe this plays a role. We can test that in mice. So then there's mouse models that overexpress alpha-synuclein, or where you actually inject alpha-synuclein fibers into the brain and create a mouse model of Parkinson's. Now, it's not correct to say the mice have Parkinson's disease, but they have increased levels of alpha-synuclein, which is one of the core features, and they develop loss of the dopamine-producing neurons, just like in Parkinson's disease. So the hope is then we can, in these mice, test strategies that work to protect against this alpha-synuclein toxicity, and then go back to the humans and test these in humans and see if they work. And we've had lack of success with some of the other neuroprotective agents, but they weren't all tested in alpha-synuclein-based models. Some of them were tested with acute toxin models that aren't maybe not as good of a predictor of success in humans. We don't really know that yet, but we'll see in the future. Maybe we'll start seeing better success in part because we have better animal models. So here's a couple examples where immunotherapy has been tested in the mouse models. This is one from Virginia Lee's group where they have a model where they make fibrils of alpha-synuclein and then inject those directly into a mouse brain. And what you see here by these dots throughout the brain is that it spreads. It doesn't just stay where they inject it. The pathology spreads throughout the mouse brain, and these mice have early death and other deficits. And they showed that if they give alpha-synuclein antibodies, so antibodies that are binding to and helping to clear away alpha-synuclein, that greatly reduces the levels of alpha-synuclein, including potentially abnormal forms. And here, if you just count the number of neurons in dopamine-producing neurons, it shows you that although normally next to the on the side where they inject the alpha-synuclein, you lose neurons. When you give the antibodies against alpha-synuclein, it prevents that. So that's promising. It helps block the spread. And here's another study. This is from another group, Games et al., and they have mice that are genetically modified to increase the amount of alpha-synuclein that they express in the brain. And they also develop pathology that's somewhat related to Parkinson's disease. And in these mice too, if they give alpha-synuclein antibodies, they show, here you can see, actually, does this pointer work maybe? Oh no, that's just the, there we go, sorry. So here you can see the dark pigment here is the alpha-synuclein staining, and you see a lot less of it here, and that's because in this case, the mice were given antibodies that target the alpha-synuclein, so it helps to prevent the spread of the alpha-synuclein. And in these mice, they also showed that it improved some measures of cognition and of motor performance. So now you're not only clearing away the alpha-synuclein, or at least preventing it from spreading or reducing the spread, you're also having benefits, which is important when we talk about the potential negative effects of clearing it away. Well, here it is in a mouse model, an actually living organism, that you see benefits regarding motor performance and cognition. So this brings me to another one of my favorite cartoons. Well, we've cured every monkey and rodent illness. I guess we should move on to humans. But it's not so easy, right? I mean, we actually have, there's a lot of illnesses we've cured many times over in mice. And the next slide is sort of a reminder of probably the biggest reason. That's for any Harry Potter fans in the audience. That's Peter Pettigrew. But Peter Pettigrew is an exception. Most of us are not rodents. And so the success in rodent models, while promising, and it's almost a necessity before we move on to clinical studies, they don't necessarily tell us for sure, at least certainly not for sure, that it's going to work in humans. And that comes back to the other comment that was made earlier. We will never be able to make progress towards better clinical therapies without involvement of patients, of people with Parkinson's disease participating in these trials. So that's very critical and appreciated. So I need to talk about a few problems for immunotherapy. In the Alzheimer's field, there's a way to image the protein that accumulates. So in a living person, you can do just a non-invasive imaging study and see how much of this abnormal protein, in this case amyloid, is accumulating in the brain. And you can see this color-coded image there on the left of a person with Alzheimer's disease showing a lot of the protein and much less of it in a control person of similar age. In Parkinson's, the abnormal protein is alpha-synuclein, and I just have a question mark there because there is no good imaging agent. There's a lot of candidates. They all have limitations, not specific enough for alpha-synuclein or other problems, but there's a huge effort towards identifying a way to measure alpha-synuclein in the brain, because now in immunotherapy trials, if we give these antibodies, we can't say for sure if we've really cleared it away in the brain in humans. We can in mice. In humans, we'd like to know, but we don't have the imaging study yet. So I think that'll be a real game changer when it eventually is developed. So I'm going to talk a little bit about a few of the actual studies here that are ongoing for immunotherapy in Parkinson's disease. There's one that's an active vaccine and two others that I'll mention that are involving passive immunotherapy, and I'm going to talk on the next slide about what that means. There's many other strategies that may help to reduce alpha-synuclein, nilotinib being one, and many other different strategies that are in the works, some in clinical studies, but today I'm focusing on the immunotherapy. So active vaccine means you actually give a shot that induces the person's own immune system to create antibodies against alpha-synuclein. This has some passive vaccine is where you actually just directly infuse the antibodies themselves. The body isn't making the antibodies, you're giving the antibodies. And there's pros and cons to each. For an active vaccine, it's much easier. It's easy to go in and get a shot, even if you have to get it a few times. Very quick, easy, and then your body goes ahead and makes the antibodies yourself, so it's long-lasting, much less expensive than the immunotherapy, giving the antibodies directly. But you have limited control over what the patient gets, right? Once they start making their antibodies, they'll make whatever antibodies they make, and they'll make it at whatever dose they make. That may vary person to person, and you don't really have control beyond how often you give the shots or booster shots. For passive vaccine, it requires periodic infusions, That's not so great, and it's very expensive, which actually makes me almost worried. I sure hope these work, but it's going to be hard to scale that up to 1 million people in the U.S., let alone around the world, if it does work, though it would be wonderful proof of principle that maybe might give you hope the active vaccines will work. But you do have the advantage with the precise control over exactly what type of antibodies you produce and how much and how often you get them. So, maybe at least as a proof of principle experiment to show that immunotherapy could work, it's reasonable to test that. So first I'll talk about this epitope experiment. This is an actual vaccine. It's not, they give a synthetic form of alpha-synuclein or alpha-synuclein mimicking peptide. And when they inject that in, along with other agents, it promotes the immune system to start producing antibodies against that protein. And there's some data recently published there were actually 2 phase 1 clinical studies. And one of them, as an example, had 36 patients treated with low or high dose of the vaccine or placebo, and they got 1 shot per month for 4 months and then a booster shot at 36 weeks. And the conclusion of that was, this is early phase, not going to tell you did it slow progression, but it was safe and tolerated. So that's encouraging. And equally encouraging is they did succeed in demonstrating it did induce the production of the antibodies that they wanted to. The kind of antibodies that we saw in the mouse models look very promising for having beneficial effects and clearing away the protein. But we need to move on to the larger placebo-controlled phases to really have enough power to be able to tell if they work. And by work, I mean clinical efficacy, actually slowing progression. Another study that's going on right now, one of the 2 with the passive immunotherapy has the acronym the Pasadena study, and this is Roche Prothena. This one is a phase 2 study that's ongoing now, actively enrolling, and it's enrolling 300 patients with early Parkinson's disease. In this case, early means within 2 years of diagnosis, and only people not yet on medications, although they do allow selegiline or rasagiline, but no other medications for Parkinson's. The reason we do that is if somebody's on medications, it can mask the clinical symptoms. I mean, that's good, the medications work, but it makes it hard to tell if your immunotherapy has slowed progression, because since we don't have a way to measure alpha-synuclein or count neurons in the brain of a living person, we go by the clinical exam to tell, and the meds can mask the clinical findings. So and also we think early treatment might have the best chance for success, because even by the time a person has been diagnosed with Parkinson's, probably more than half of the dopamine-producing neurons have already died, and we're not going to bring those back with immunotherapy. We're only going to prevent further loss. So if it works, the earlier you start it, the better. So that's another reason to start early. This targets what they hope is the pathogenic form of alpha-synuclein. So they're not trying to clear away all alpha-synuclein, just these aggregated forms. And this trial involves low dose or high dose of the antibodies versus placebo, IV infusion every 4 weeks. And they do that for a full year. And actually, they're even going to do a 1-year extension to go out to 2 years. Main outcome is going to be change in the UPDRS. UPDRS is this clinical rating scale where we measure tremor, stiffness, gait, other features, and come up with a score, quantitative score, for how you're doing clinically. And the hope is, we know there's a tendency for that score to worsen over the course of a year, and hopefully we'll see less worsening in people who got the immunotherapy compared to people who didn't. We're also going to look at that DAT scan that you saw as a more objective measure of dopamine uptake in a part of the brain that's involved in Parkinson's, although there's still some uncertainty over exactly what that measures. So we really need to, both the clinical part of the study is important too. Just a little plug for anybody who's interested. We are one of the sites recruiting for that. So if anybody's interested, I'm not the person running the study at our site, but I'd be happy to put anybody who's interested in touch with the folks at our site who are doing it. Next study is, the next study is being run by Biogen. This is one, actually, I am involved on the steering committee, but we're not a site, but Massachusetts General Hospital is a site, so this is another option. This one is also a phase 2 study looking at 311 people with early Parkinson's disease, in this case defined as being diagnosed within the past 3 years, but in this case you can't be on any medications for Parkinson's disease. And like the other study, this one has an antibody that they believe targets just the pathologic form of alpha-synuclein. And there's 3 doses of their antibody being tested versus placebo every 4 weeks for a year, so that part's similar. And in this case, the main outcome is just safety and tolerability, as is often the case for phase 2 studies. Secondary outcome, like the other one, is going to be the change in the DAT scan, the imaging. And for either of these studies, if they work, hopefully then it would move on to a phase 3, which should be like the inosine phase 3 study, the definitive study to tell us yes or no this works and should we be doing it in patients. So very promising and already well into the phase 2 clinical studies with multiple studies ongoing, but there are potential problems. One I've already mentioned, mice aren't humans, that's a problem. Lack of biomarker for alpha-synuclein, no good way to measure it. That's another problem I've mentioned. And also the imperfect measures of progression. I mentioned we use just clinical exam, which is somewhat subjective and imperfect and influenced by a lot of things besides how many neurons they have in there that produce dopamine. I mentioned phone apps here because there's a potential to get more objective, quantifiable data from simple phone apps like that allow you to tap and test tapping speed, and people can do this at their home. They could do it, once a week, every day even. It makes it and actually this is now being done as a sub-study in the inosine study as well. There's still need to figure out which are the best apps and how well they really predict how good they are at markers of progression, but there's ways to get better at being able to mark progression so that we can have better power to detect a successful neuroprotective agent when we finally have one. Another problem that you've heard is that the diagnosis isn't always certain, and that's especially true with very early patients. So some of these patients who we diagnose with Parkinson's and enroll in our clinical study don't actually have Parkinson's, and they might not have any potential to benefit from the treatment that we're testing, and that will reduce our power in the overall study to detect an effect. One way to reduce that is with DAT scans that can, and you would not enroll a person if they clinically look like they had Parkinson's, but their DAT scan is normal. Of course, that abnormal DAT scan doesn't prove it's Parkinson's because some of the other things that can mimic Parkinson's also have abnormal DAT scans. But it helps. It helps to improve the accuracy. Another potential problem, I pointed out a couple of times that these studies, at least the passive immunotherapy studies, are trying to target the pathogenic form of alpha-synuclein, but we don't really know for sure what is the right form that should be targeted. Maybe we're not targeting the optimal conformation of alpha-synuclein. There's other basic facts we don't know. How long should we be treating? How long do you have to treat to be able to detect the effect you're looking for? And are we starting too late? I mentioned the studies are taking early Parkinson's patients, less than 2 years or less than 3 years, but that's already well into the illness pathologically. Maybe you need to start a couple of years before the diagnosis. You might think, well, how can we do that? How can we enroll people who haven't been diagnosed yet? Well, there are potential ways. One, for example, is REM behavior disorder, which is an abnormality where people tend to act out their dreams. And people who have REM behavior disorder have a high risk over the subsequent years of developing Parkinson's disease. So maybe we need to even go earlier. For example, try our therapies in people with REM behavior disorder and see if we can delay or prevent conversion to Parkinson's. But that's not been done yet here. And the last thing I want to talk about are the trials from Alzheimer's disease. Because I think the immunotherapy trials to me look extremely promising, but I have in the back of my mind, well, how did I feel about the Alzheimer's trials before the results came out? In Alzheimer's trial, there were several similar trials in Alzheimer's disease targeting the protein, one of the proteins that accumulates in Alzheimer's called amyloid, beta amyloid. And here's one publication describing 2 phase 3 studies. These were huge and expensive and complicated phase 3 clinical studies. And here's the key line from the abstract. They didn't work. They did not meet their primary endpoint for clinical efficacy for either of the studies. And there was another study of a different antibody also targeting amyloid, and it had no effect on cognitive decline. So that was quite disappointing, as you can imagine. This was a huge effort and agents that did seem to engage the target, amyloid, but didn't have the hoped for clinical benefits. So why did they fail? Well, one of the biggest reasons, at least that some people think, and hope is that they started too late. And I say and hope because there's great effort now, partly led by Risa Sperling, who's at Brigham and Women's, and other people, to start these antibody treatments even earlier. In the case of Alzheimer's, they have the advantage of that non-invasive imaging. So she is now leading studies to look at people who are elderly people who are cognitively normal, but by imaging already have amyloid accumulating in their brain. So they might be pre-symptomatic Alzheimer's patients, and you treat those patients with the antibodies and see if you can delay or prevent them from clinically developing Alzheimer's disease. So there is a way to go even earlier than just recently diagnosed. And that's in the works. Another possibility in the Alzheimer's field is that maybe it's the wrong protein. There's a debate in the field over exactly what role does amyloid play, and is targeting amyloid the right one? Maybe there's a different protein called tau you should target, or maybe you can't target amyloid alone. Maybe you have to target both. In Parkinson's, I think this is somewhat less controversial. There's pretty broad agreement that alpha-synuclein plays a critical role, though there's, as I said, still the uncertainty over which form. But I don't think there's much controversy over the idea that alpha-synuclein is important in Parkinson's. I mentioned the confirmation issue. There's other maybe technically, somewhat technical issues, but these are critical to success or failure in a study. Maybe you're completely on the right track and it just wasn't the right dose. Or again, with Alzheimer's, they have the problem of incorrect diagnosis. Even if a person has amyloid, there may be other pathologies as well, such as vascular disease that can contribute to the dementia and make it less easy to detect a response to the treatment. But as I mentioned, further studies are underway in Alzheimer's despite these initial very disappointing results. So quick summary then, the good and the bad on immunotherapy. On the good side, alpha-synuclein is clearly a relevant target. The studies are starting at early stages, which is the best way to do it. And it's clearly effective in mouse models of Parkinson's disease. And the early phase results that we have so far do show at least that it's safe. But, we're not sure which form of alpha-synuclein should be targeted. We can't measure whether or not we're actually targeting, clearing away alpha-synuclein in the brain, in the patients in these trials. That's what we really want to do, and we have no way to prove that in the patients right now. And as I mentioned, even though it's early, we don't really know that it's early enough, and the mouse models don't predict success. So back to the slide then. What do you think? Is it hype or hope? I'd say I wouldn't call it hype, because I think if you say, is there a potential, I wouldn't use the word cure, but for a dramatic change in the way we treat Parkinson's, I'd say yes, there's a possibility here. But am I ready to put all my money in that one basket? No, because it might be a home run, or it might be a pop fly, out, and we just don't know yet. And we have to do the studies to see. But thankfully, it's not the only way that we're looking at this. I like this Yogi Berra quote, which I twist around in a more positive sense. So I've been talking about immunotherapy here as a potential way to achieve disease-modifying or neuroprotective therapy in Parkinson's. But this, again, a partial list of just many other strategies, some of which do involve alpha-synuclein, some of which involve other mechanisms. But there's a lot of very promising strategies, and many, many more that could go on the list at even earlier stages of development. And I think it would be too optimistic to say they're all going to work, but I think it would be too pessimistic to say they're all going to fail. I think we're making a lot of progress. We're having a better understanding of the precise molecular targets we should be going after, better mouse models, so hopefully more predictive of success. And as we'll hear from Peter Lansbury in a minute, and we heard from Peter, in some cases, much more targeted therapies for Parkinson's subtypes. So I'm still very optimistic, and I'll just end on this one slide. Just also as a reminder, there's a lot of other things besides just thinking of the future, as Michael said. We have wellness programs. I'm a big proponent of exercise, and Michael showed you some of the reasons why. And participating in clinical trials and education are other things you can do now. So I'll stop there, I guess, take a couple of quick questions. Are we just going to go people asking rather than written this time, or? Okay, sure. Yes.
Are there risk factors for study participants?
Well, yeah, I mean, any study has certain inclusion and exclusion criteria, some focused on safety, but the most restrictive criteria is the need to not be on symptomatic medications and to be very early in the illness.
And any risks for participating?
Yes, there's always risk, and anybody who, if they were interested in participating in this, there's a very detailed, many-page informed consent forms that goes over in great detail all the potential risk. And one of the risks to keep in mind always is that we may not know all the risks at this point. Sometimes that's one of the reasons we may learn about problems we don't know. But like I say, for the most part, the early studies did suggest in the smaller number of subjects that it was reasonably safe and well tolerated. But yeah, we can go. Anybody who's interested can learn in more detail about those. Yes.
Well, it seems that most Parkinson's patients get diagnosed, and then with hindsight, they look back and they see, oh yeah, 3 years ago, oh, 4 years ago, these things were going on. And so it seems like these possible therapies will never get to the Parkinson's patients basically in the room who have it, who are on Cinemet, because you don't know you have Parkinson's right away. It doesn't show up necessarily as a tremor, and who knows how long other things were going on. So it seems like it's geared towards, I don't know, the basic population.
Yeah. So just point out that a few points that Parkinson's patients have symptoms maybe a few years before they're diagnosed in some cases. One point I want to make, first of all, of course, you're right. We tend to use the diagnosis rather than date of onset as a criteria for the study, in part because it's probably more precise. Sometimes somebody will tell me, well, I've had the tremor that started a year ago, but I think I wasn't swinging my arm for 5 years. And so is it 1 year ago or is it 5 years? I don't know, but usually we can pin down the date of diagnosis, and that's not perfect either. Some people may tolerate mild symptoms longer than others, but we have to choose objective criteria. Even though we choose early patients not on medications for some of these trials because it makes it easier to detect an effect, in theory, if these work, I don't think the treatment for patients will only be restricted to those not on medications. The hope is they would be applicable and beneficial to others. Just for the purpose of being able to detect the effect, it's better to do it in clinical trials. And also, it's true that those who, even though people at later stages may still benefit, those with the most to benefit would be the earlier stages. So, and I did mention about REM behavior disorder as one example of how we might be able to find people even before diagnosis. And there's other ways, population screening could be done to pick up early signs, or maybe if there's a better, easy to do imaging study, like with the amyloid imaging, you can find people who are pre-symptomatic. Should we maybe take one more question? Yeah. Yes? Yeah. Yeah. Yeah, its question is about FDA policies and what could the FDA do to speed things up. It's always a balance with the FDA between wanting to do things as fast as possible to get the treatments to people who need it now or as soon as you can, and not wanting to cause unnecessary delays, versus two factors that run counter to that. One is safety. If you go fast, you might not pick up on toxicity, and that's happened. Actually, tolcapone might be one example of a drug that was FDA approved, and only after it was approved was it discovered that it could cause serious and even fatal liver toxicity. But thankfully, it was subsequently learned with proper monitoring, you could easily mitigate that risk, and it was brought back on the market. But it was essentially something that wasn't recognized earlier on. And a second factor is if you do relatively limited studies and approve something before it's really definitive, you could easily get drugs where patients believe it works, but we really don't know for sure, and we don't want to have FDA-approved drugs on the market that we're really not sure they actually work. Placebo effect is always a factor to consider, and even when patients feel it's better, we need to know more than that. We need to know that it really is better than placebo and that it's safe. So it's a tough balance between those different factors. For which one? I didn't. Yeah. Yeah. Hopefully that's a rare side effect. But yeah. Good. Yeah. So yeah, it's a very complex issue. A lot of factors need to be considered for the FDA decisions. I think just because of the time, we're running a little late. I should let the next speaker move on. So move on to the next speaker.